I am Bernard Herschel. Before my retirement, I was the chief of HIV/AIDS at the Geneva University Hospitals. You may call me an AIDS dinosaur who has followed the HIV epidemic from the very first cases in the early 1980s through the triumph of antiretroviral therapies, a triumph unequaled in the annals of medicine. This triumph has its flip side. Younger colleagues are no longer familiar with the opportunistic diseases and the myriad manifestations, hence the idea of these videos. After a short introduction, they will show a series of images illustrating one or several exemplary cases from my files. What did I think when I read the first reports of what turned out to be AIDS in June of 1981? Not much, I'm afraid. I remember a hastily arranged special session at a large infectious disease meeting in Chicago in October of 1981. Much head-scratching and bafflement, some feeble jokes, not always in good taste, and hopes that something as crazy as a gay plague would pass us by. This type of reaction was widespread. One could quote, for instance, the World Health Organization, which considered AIDS to be a problem of overindulgent Westerners and an unwelcome competitor for funds that should rightfully go to more important problems like tuberculosis and malaria. Or the French philosopher Michel Foucault. How perfect! You American Puritans keep on inventing diseases, and this one kills only blacks, drug addicts, and homosexuals. Really perfect. It took a year or so to realize that this was no laughing matter with the first patients in Geneva. Mrs. B was a 48-year-old housewife who presented in May 1982 with weight loss and oral candidiasis, and then in June with anemia, fever, and pulmonary infiltrates due to pneumocystis pneumonia, the archetypal AIDS-defining disease. Treatment was ineffective. She died in August. But what was the cause of this enigmatic disease, which very obviously killed people with no relation to gay men in the USA? Hypotheses abounded, all unlikely. For instance, drugs, especially amyl nitrate, sperm being released in the blood followed by an autoimmune mechanism to destroy T cells, antigen overload linked to quote-unquote lifestyle, multifactorial, which usually means we have no idea, a fungus producing cyclosporin, Epstein-Barr virus, cytomegalovirus, a virus escaped from a biological warfare laboratory, African swine fever virus, because you know there was a CIA plot to kill all the pigs in Cuba, but something went wrong, etc., etc. Enter HTLV, the human T lymphocytotrophic virus. In 1977, Japanese scientists had postulated that the virus must be the cause of a rare form of leukemia, and in 1980, the virus, later called human T lymphocyte virus, was isolated by Bob Gallo et al. This virus infected the very cells that had disappeared from the blood of patients with AIDS, and like AIDS, was transmitted by IV drug use, sex, and from mother to child. It was a much better candidate than all this nonsense about sperm and autoimmunity and antigen overload and pig virus and the CIA. By 1982, many people were convinced that this or something like it must be the cause of AIDS. One cannot understand the controversies around the discovery of HIV without considering the personalities of the two main protagonists. Bob Gallo was already well known before AIDS because of his discovery of IL-2, which allowed the culture of T lymphocytes, and because of the isolation of HTLV1. And he was a brilliant speaker. Have you ever, at the end of a scientific talk, thought, too bad this is already over, I'd like to have listened another 20 minutes? This happened to me exactly once in 50 years, and the speaker was Gallo. His collaborators thought the world of him. It didn't hurt that he had the allure of an Italian macho with a beautiful wife, and, so the rumor went, an even more beautiful mistress who worked in his lab. Once a year he organized what he called a lab meeting, actually a scientific conference with some 500 listeners in attendance, where from morning to night one 15-minute talk followed another, from genetics to oncology to pharmacology to physiology and back, with Gallo in the front row, who peppered the speakers with questions. Survivors were invited to a dinner where Gallo's friend Francis Ford Coppola, the filmmaker, made a moving speech expressing his hope that science would find a cure for AIDS. Not so Luc Montagnier. To begin with, many of his collaborators were not shy in stating that they held him in low esteem, both personally and scientifically. And he had a reputation as a terrible speaker. To prepare the International AIDS Conference in Geneva in 1998, there were all those bodies euphemistically called organizing committees. They mostly fought each other, but they agreed on one thing. Don't you dare invite Montagnier as a speaker. He rambles on and on with this terrible accent so you can't understand a thing, and what you understand is nonsense. Like mycoplasma is responsible for the damage AIDS causes, and water has a memory. So what to do? How to tell him? The solution we found was to film a video. We showed up with half a truck full of equipment, a professional interviewer, and a makeup artist. It was expensive, but enough of a show to satisfy any ego. And although his 40 minutes of mumbling were cut to 4, Montagnier was happy. And AIDS was big news then. Here is a sample of some of the headlines from the mid-1980s. Bob Gallo in particular was much appreciated by the media, like at this press conference in April of 1984, when the US Secretary of Health, Margaret Heckler, announced the probable availability of an AIDS vaccine within a couple of years. Here is another example. The first big AIDS conference in Europe took place in Brussels in November of 1984. I was walking through the old town in the evening after the opening ceremony and came upon a crowd of people who were obviously looking at something exciting, with a truck marked Radio Télévision Belge Francophone RTBF and camera and powerful lights being set up. And what was the sensation they were trying to film? It was Bob Gallo eating mussels and French fries in a restaurant. That was the sort of atmosphere in which the controversy around the discovery of HIV unfolded. Back to May of 1983, when these two articles appeared in Science. Isolation of human T-cell leukemia virus in AIDS from the Gallo group, and isolation of a T lymphotropic retrovirus from a patient at risk for AIDS by the Montagnier group. Gallo et al. Reported isolation of HTLV of one AIDS patient and evidence for HTLV sequences in 2 out of 43 AIDS patients. Montagnier et al. Isolated from one patient something that looked like a retrovirus in the electron microscope, had evidence that this was a new virus antigenically distinct from HTLV. What to make of this? Everything one knew about the investigators involved and about the resources they commanded pushed one to go with Gallo. But 2 out of 43 patients, and these were immunosuppressed with AIDS, likely to pick up any kind of virus from their surroundings. The credibility of both groups now hinged upon isolation of the putative causal virus from more patients and or showing that AIDS patient displayed an immune response against this virus. Patient X was an employee of an international organization who had spent years in East Africa. In January 1983, he received a diagnosis of ITP, idiopathic thrombocytopenia. In April-May of 1983, he had candidiasis and weight loss, and in August, encephalopathy with impaired consciousness leading to a brain biopsy. The brain biopsy yielded a diffusely infiltrating tumor made up of immature lymphocytes. The diagnosis was a primary brain lymphoma. Malignant cells were also found in the cerebrospinal fluid with mitoses and prominent nucleoli. In the spring of 1983, we sent a serum sample of patient X to Françoise Barré-Sinoussi for immunoprecipitation. After 2 months, there was still no result, so I called. And she said, "Oh, this may be the positive sample where the label fell off and we couldn't figure out where it came from. Can you please send us another one?" We did, and the result was indeed positive. What you see here is immunoprecipitation of the radio-labeled viral P24 protein by the serum of patient X. This was better than nothing, but immunoprecipitation is too complicated for routine use. In early 1984, both the Gallo and Montagnier labs were developing enzyme-linked immunosorbent assays, or ELISAs. We are now in early 1984, and in Gallo's lab, yet another virus labeled by Gallo HTLV3 was isolated from an AIDS patient in San Francisco. At a press conference on April 28th, Margaret Heckler, the US Health Minister, and Gallo presented the result. She said, "In particular, credit must go to our eminent Dr. Robert Gallo, who directed the research that produced this discovery. I would like to turn now to the eminent scientist who has made the breakthrough which prompts the conference today, upon Dr. Robert Gallo." Note, no mention of the French. And Gallo said, "In our group, I have to mention someone who came to us from Czechoslovakia, Dr. Mikuláš Popovič, who played a very major role in some of the growth of cells that would be permissive for the large-scale production of the virus. Also a postdoctoral from Switzerland, Dr. Jörg Schöppach, etc. Keep these two names in mind. We'll come back to them. Jörg Schöppach, who worked in the laboratory of Bob Gallo in 1984. Which is one reason why the ELISA test used to diagnose HIV infection was based upon serum samples from Swiss subjects. But I'm getting ahead of myself. This was 1985, and we are still in the spring of 1984. When a number of refugees fled from Zaire to Switzerland. I don't remember how and why, but I got hold of about 200 anonymous blood samples from these Zairean refugees and called Jörg Schuppach to ask him whether he would be interested to try his new tests on these samples. He was interested. It so happened that my wife and I planned a holiday in the States. Flore, my lab technician, made a nice package with lots of dry ice. I took this with me on the plane to the States. Somewhere over the North Atlantic, I noticed what looked like smoke drifting out of the overhead bin where I had put the package. I knew that this was just cold air from the dry ice, but somebody else might be worried, so I placed the package under my seat. It cooled my backside for the rest of the trip, very uncomfortable. We arrived and went through customs. Well, I've been to the States maybe 40 or 50 times and nobody ever was interested in the contents of my luggage. Except this time. The customs agent eyed the package and asked, "What this?" I was prepared and said, "Biologic samples for the National Institutes of Health without commercial value." He laughed. "Not AIDS by any chance?" "Oh no," I said, and through I was to the DHL office and off to NIH went the package, whereas Suzanne and I departed for the splendors of the American West. After this nice vacation, it was back to the hospital, and I more or less forgot the package at the NIH until August, when I found my lab technician Flore in tears. The results had come back from Jörg Schuppach, and 11 of the 200 samples had tested positive. But why was Flore crying? Unbeknownst to me, she had a boyfriend from Zaire. She had taken his blood and added the tube to the package, and his sample was positive. Luckily, this story had a happy ending. In the meantime, my colleague Luc Perrin had set up a blood test which was different from the ELISA, called Western blot. Here you see several of these blots. The test is positive when there are black lines. On the left, a positive sample from patient X, the one that was already positive in France a year earlier. The lanes labeled 19, 21 and 23 are other positive samples. The lane labeled D is from Flore's boyfriend, Diwani. It is negative. What conclusions can we draw from this episode? Well, quality matters. Homemade tests, even concocted by brilliant researchers, are not enough. The first versions of Gallo's, Abbott's, HIV tests were plagued by false positive results, particularly in multiparous women and Africans. There was an initial distrust towards the screening of blood transfusions, but that was happily non-justified. HIV tests are now among the most reliable of all tests in medicine. X and D continued and end. Mr. X died in 1983. Mrs. X fell ill in 1990 and died from AIDS-related dementia in 1992. Diwani and Flore married and had 3 children, one of which was a classmate of my daughter in medical school. At the end of 1985, the situation appeared clear enough. Montagnier had evidence for a new virus isolated from patients with AIDS or pre-AIDS in France, and this virus was called LAV subscript Bru. This finding was at the origin of a diagnostic test developed by the Pasteur Institute. Gallo isolated a more or less identical virus from an AIDS patient in San Francisco, called it HTLV3 subscript SF, and grew the virus in large quantities. Based on this, Abbott developed another diagnostic test. The genetic sequences of LAV Bru and HTLV3 SF were virtually identical, and this presents a problem to which we'll come back. The controversy about the discovery of HIV sprang back to life after having seemingly disappeared, and a couple of patients treated in Geneva had something to do with this. They were husband and wife, opponents of Mobutu, and refugees from Zaire, where they were imprisoned. In 1982, both had diarrhea and relapsing fever, serious herpes infections, encephalopathy with AIDS-related dementia, and died within days of each other in January of 1984. This flowchart summarizes the couple's history. Among other complications, both had Salmonella typhimurium bacteremia. Without quinolone antibiotics, which were not yet available in 1983, it was impossible to eradicate this infection. We plasmid typed these Salmonella typhimurium by isolating their plasmids and then digesting the plasmids with a restriction enzyme. As you can see, the two isolates are strictly identical. To the controversy. Here again is a timeline. In May of 1983, Montagnier et al. Described the isolation of LAV Bru from a patient named Brugière. In July and September of 1983, samples labeled LAV Bru are given to Gallo by Montagnier. In April of 1984, Gallo describes the isolation of HTLV3-SF from an American patient, SF standing for San Francisco. In early 1985, the sequences of HTLV3-SF and of LAV Bru are found to be identical. From 1985 to 1990, as information about HIV accumulates, it becomes clear that isolates vary. If the sequences are identical, it follows that they must originate from the same sample. How is it then possible that a virus isolated from a patient in France is identical to a virus from another patient in San Francisco? Things came to a head in 1990 when the American journalist John Crewdson published a violent attack against Gallo in the Chicago Tribune and later in a book, Science Fictions, A Scientific Mystery, A Massive Cover-Up, and the Dark Legacy of Robert Gallo, accusing him, among other things, of having stolen the virus of Montagnier. An inquiry by the NIH revealed that the isolation of HTLV3-SF resulted from putting into the same culture different viruses from different patients. Among these viruses were the two MOVs. Popovich had obtained these samples from Professor Maurice, Chief of Oncology in Geneva. Popovich's notebooks refer to the resulting isolate alternatively as LAV, HTLV3, and MOV. In 1991, we received a request by the NIH. Do you still have samples of MOV? These were needed for sequencing and comparison of all the viruses put into the quote-unquote Popovich soup. This opened the possibility that the most famous isolate of HIV came from Geneva. Here is what Popovich had done. He had taken samples of several patients, 4 of which are shown here. A sample supposedly from Frédéric Brugière from France, a sample of Mrs. and Mr. Mauve from Congo by way of Geneva, and a sample from a patient in San Francisco, and put them into the same culture, which I'll call the Popovich soup. These viruses were all different, as shown by the different colors. A couple of weeks later, the soup was full of virus, but what virus? Is it Brugière's, MOV 1s, MOV 2s, or SFs? In the lab book, Bru, MOV, and SF are all mentioned. Finally, Gallo calls it HTLV3-SF. HTLV, I suppose, because he still hoped that it will turn out to be a variant of the virus he was among the first to discover, and SF because of the supposed origin. But what was the result of the reanalysis by the NIH in the early 1990s? Well, there was a big surprise. The virus that grew out was unlike any of the viruses that were supposedly put in. It was like throwing potatoes into the kettle and getting pea soup out. The explanation lay in what happened in Paris back in 1983. You already know about Bruguière's virus, published in Science in May. In June 1983, the Montagnier group isolated another virus from Christophe Layet, which grew faster and better than other isolates. It overgrew other cultures, both in Montagnier's and Gallo's labs. When Montagnier gave Gallo a sample of what he thought was LAV Bru in July of 1983, it was actually LAV Layet. And when his and Gallo's group sequenced the virus in early 1985, they were all sequencing the same isolate, namely LAV Layet. In conclusion, Popovich and Gallo have re-isolated a virus provided by Montagnier. The virus called HTLV3-SF by Gallo was neither HTLV nor from San Francisco, but from a French patient called Layet. And later, Mr. and Mrs. Mauve have not had post-mortem glory. Gallo and Popovich received a censure by the NIH in 1992, which was however cancelled in 1993. The financial arrangements between Abbott and Pasteur were revised in 1994 with more money, $6 million, going to Pasteur. And here's a question for you. Is it correct to say that Robert Gallo is a co-discoverer of HIV? The Swedes didn't think so in 2008, when they attributed the Nobel Prize to Françoise Barré-Sinoussi and Luc Montagnier, with no mention of Robert Gallo. Thank you for watching, and thanks in advance for your feedback. This video is part of a series on HIV/AIDS. If you are interested, please subscribe or click on the latest upload top left.